Hello, everyone. Good afternoon. Thank you for coming to this presentation. I am Tofi. My name is Tofi Soneye, and I am a rising sophomore at Fisk University. I am a double major in biochemistry and data science, and I have been very interested in bioinformatics right from when I knew that I could intersect the knowledge from biology with computer science. So this summer, my project was on identifying vascular disease and cell type specific transcriptomic signatures in diseased and healthy arterial tissues. I worked at the Segre lab in collaboration with Dr. Raj Gupta of the Brigham and Women's Hospital. So what motivated our project was the desire to understand the cell and gene types that play a causal role in vascular diseases. And these vascular diseases are those which affect the circulatory system. So any disease that affects the arteries, capillaries, and veins is a vascular disease. So these are some of the more common vascular diseases, but the one I focused on primarily in my project was temporal arteritis. Temporal arteritis is a disease caused by the inflammation of the temporal artery, which is located on the side of the head. It usually presents in patients that are between the ages of 50 to 70 years. And in this project, we tried to analyze the cell and gene types that play a causal role in temporal arteritis. So we obtained data from two data sets. One was from the thoracic aorta. Here we had a total of five donors, of which three were diseased and two were healthy, which served as our controls. And then from the temporal artery, which as I mentioned earlier, is located at the side of the head. Here we had 6 donors in total, and half were healthy while half were diseased, had the disease status. So these are some of our research questions. And here in the aorta data set, we already had the gene markers, while the temporal artery data set was part of our analysis was understanding and using these gene markers to determine the cell types in the temporal artery data sets. So one of the preliminary questions was just to compare the two cell types and see the differences and similarities between them, especially using the harmonization of the single cell RNA data. And then another question we asked was what are the genes that are differentially expressed in these cell types between disease and healthy? And here we have the third question, which was basically to determine if the expression of the genes that are genetically associated with the vascular diseases enriched in specific cell types in temporal artery, because finding that results would help us know if pathogenic cell types for vascular diseases were present in the temporal artery dataset. So here is just a violin plot of the quality control metrics. Usually when you're working with analyzing single cell RNA sequence data, the first step is usually to observe the data set and really determine what quality control metrics you would use to filter out some of the things you wouldn't really need to ensure more accuracy. So based on these plots, these preliminary plots that gave me an idea of what the data looked like, I filtered out the outliers, which were the cells with less than 200 genes and more than 99.7 percentile N count, gene and read counts. So this was done to ensure data reliability and accuracy. And then here is just a histogram of the cell type numbers in the aorta data set before and after filtering. Here we can see that the VSMC1, which is the vascular smooth muscle cell 1, was the most affected by the filtering, which means it had the most cells with less than 200 genes. And here is the same plot for the temporal artery. Here we can see that not many of the cells were so affected by the filtering. It was mainly the NKT cells, which are the neuronal killer cells. They're a form of immune cells. So here we have clustering. Now clustering, I did this clustering with, after running preprocessing steps like, what? Hello?
You're okay to continue.
Okay, I did this this cluster after running some preprocessing steps like normalization, scaling, principal components analysis, and log transformation to reduce the dimensionality of the data and give more accurate results. So here is a cluster of the aorta cell types. If you notice here, this basically shows us the distinct cell populations, and this is the same chart, but for the same graph, but for the temporal artery data set. Now, if you notice the difference between these two, you observe that the temporal artery data set has the clusters listed as numbers, while the aorta data set has the clusters listed as names. So this was because Dr. Raj Gupta of the Brigham and Children's Hospital, in his lab, they had already done the clustering and identification of the aorta cell type, but the temporal artery data was relatively new, and we still had to do a lot more discovery of what it actually contained. So this actually drove us to run into harmonizing between the two data sets. We used harmony, which basically involves integrating between two data sets and plotting them on a certain plane where you can compare both accurately. And here we used batch correction to correct for sample ID and disease. And batch correction basically removes technical variation and essentially drives at more accurate results. So here we were able to determine how the cluster numbers, we're able to see how the cluster numbers aligned and overlaid nicely with the cluster names that we already had. And it was actually through this harmonization that we discovered an 8th cluster, which was a third type of endothelial cell, which we called EC3. This is actually more common in mice populations than human populations, hence why we saw it here. We actually discovered that it's a lymphatic cell, while the EC1 and EC2, that endothelial cells 1 and 2, they were, the EC1 was microvascular, while the EC2 was macrovascular. And so here we just show, this is a description of the control and disease for the temporal artery samples. You know, I'm showing the disease here is giant cell arteritis because in this analysis, we were looking at the diseases that affect the temporal artery. So here we used harmony to correct for sample ID, and we were able to obtain this cluster that showed us how the cells were divided based on their disease status. So here is this is a differential gene expression table, where for the differential gene expression analysis, I ran the Wilcoxon rank sum test to do the differential gene expression, and we were able to obtain a table of the cell types and the genes that were most significantly expressed, which were the genes that had an adjusted p-value of below 0.05. So here we just have we ran it on vascular. I picked from the 10 clusters, from the 10 cell clusters, I picked 7 which were relevant. So 5 of this actually were the vascular cell types. And then I picked the NKT, which is an immune cell. It was to serve as more of a control or a test, which would help us see how accurate our results were. So here is just a table showing the cell types and the number of genes we were able to find were significantly differentially expressed with some of the gene names. And here I ran gene cell enrichment analysis which I used a tool called gprofiler for. Gprofiler just takes an input of your genes and runs it against a background of all known genes and gives an output of all the pathways that these genes are significantly enriched in. So here in endothelial cell 1, EC1, we can see that it was significantly enriched in only one pathway, actually, which was tissue modeling. But in EC2, we had more interesting results. We could see that it was enriched for cell adhesion, sprouting angiogenesis, endothelial cell migration. As you can see here, a lot of the pathways here involve various types of cell migration. And we found similar results for EC3. Here we saw that EC3, the genes in EC3 were more significantly enriched for pathways that were involved in membrane transport activity and homeostasis. And right here, the matrix boxes right here actually show us the genes that are actually driving this enrichment. Now, if you remember earlier, I mentioned that the NKT cells were somehow were served as a control to kind of point out the accuracy of our results. So here we could see that the NKT cells, which are a type of immune cells, were more enriched in pathways that are associated with immune responses in the body, like inflammatory response to stimulus, and cell activation. So for the next step here, in our analysis, our question was really that are the genes associated with vascular diseases enriched for expression in specific cell types in the temporal arthritis trait or disease. So we used this tool, which was actually developed by my host lab, the Segway lab, which is based on expression EQTL and tests for enrichment using a Bayesian fission exact test. So the ECLIPSA method basically takes the genome-wide association. It basically maps genes from the genome-wide association studies loci to the genome-wide association study and determines the cell type specific expression. And through this, we're able to determine the genes that are more significantly enriched and the traits that are more significantly enriched. Sorry. And here is a bubble map showing the genes that are primarily enriched in some of the vascular cells and the remaining cell types. Here we can see that the vascular smooth muscle cell actually had the most genes that drove enrichment in the various vascular diseases and traits we studied, like stroke, migraine, blood pressure. And interestingly, the CD40 cell, which is actually an immune cell, we could see that it didn't really have any enrichment in those traits, but had enrichment in the IBD, which is irritable bowel disease. And this points at the accuracy of our results, since the irritable bowel disease is not a vascular disease. So in this bubble map, the columns correspond to the cells, the rows correspond to the diseases, and the size of the cell, the size of the map gives us an idea of the FDR score. Sorry. Yeah, it gives us an idea of the significance. And here we were able to determine that, migraine has the strongest, VSMC2 has genes with the strongest enrichment in migraine. So here is a research summary. In this analysis, we were able to define cell types through harmonization with AOTA and Another thing we did was finding genes that change with disease status, and in addition, the cell types enriched for genes associated with vascular diseases. I personally had no experience with anything in bioinformatics, anything in R, anything in single cell RNA sequencing data. And this internship, this period really taught me a lot about processing single cell data and the importance of patience and diligence in research. I really learned to walk through my errors and find solutions, even though I tried something multiple times and it didn't work. It taught me to really go back and observe and be more careful, especially when I was running code. And here are some of the applications of single cell RNA sequencing analysis in the real world. It's something that would help us understand complex biological processes, identify potential therapeutic targets, and personalized medicine applications. This is actually my favorite point, because I believe that, you know, if we could study the genome of a certain family and be able to run single cell RNA sequencing analysis, we'll actually be able to understand what diseases they are more prone to and probably help them in adjusting their lifestyles or medications to accommodate for that. So these are some of the next steps of the project. Hopefully I will be able to do this during the extended research. So some of the things we want to do is to harmonize our data sets with the HuBMAP reference data to identify the tissue-specific transcriptomic signatures. Now, in this project was more of a small-scale version of that, because here we did the cell type-specific transcriptomic signatures, but we think that it would be good to compare tissues from the kidney to tissues from the heart, tissues from the skin to tissues from the eye, and actually determine these organotypic signatures. And another great step would be to run Eclipsar on the GWAS of giant cell arteritis and on differentially expressed genes between disease and control in the artery tissues. So here are my acknowledgements. I'm very, very grateful to the NIH and HuBMAP for this wonderful opportunity. I'm grateful to have worked with Dr. Segre. She was a wonderful, wonderful mentor to me, and I am happy that I was able to learn so much under her. I'm grateful to all the other members of our lab, like Arushi Srivastava. She supervised my project and helped me a lot when I ran into errors. And then Dr. Raja Gupta and his team for helping with the preliminary analysis and for being there whenever we had questions to ask about the data. And also once again to the NIH and HuBMAP, Maya and Van Zanten, Ajay Pillai, and Yulia Strenkalova. I'm happy that I got to meet with you at least once weekly to really just talk about the things in my project. And even when I had problems, there was always someone to ask, there was always someone to talk to. So I am very, very happy for the opportunity. And are there any questions?